Hello, my name is Byron Cheatham, and I'm Vice President at CytoViva. And at CytoViva, we're in the hyperspectral microscopy business. And our friend Neil Bogart had asked us to create a brief video talking about how hyperspectral microscopy is utilized in the food safety counterfeit analysis process, as well as for drugs. And so we created this very brief video just to illustrate how hyperspectral microscopy is used in these applications. And we just want to start by actually showing you what a hyperspectral microscope looks like. It looks like a very basic research grade optical microscope that includes some specialized optics and some specialized hyperspectral image capture and analysis capabilities on the microscope. These optics can work down to the nanoscale with the microscope. And with hyperspectral imaging or hyperspectral image capture, every pixel in the image contains that pixel's spectral response. And we just want to show you very briefly what that looks like and what you can do with it. If you look here in this image, this is a hyperspectral image of serotype, which is used as a COPD medicine, for example. And there are three elements in the serotype. There's the excipient, which makes up the primary aspect of the drug, but also there are two other active ingredients, salmeterol and fluticasone, which are in the drug as well. And if I collect spectrum here in this hyperspectral image and I go to different pixels, I can get different data sets. And you see these different data sets here. And if I turn all of those data sets on in hyperspectral imaging, I can map each of these elements in the sample. And so here we're mapping in yellow the excipient, which makes up the primary parts of the drug. This is a 10x image, hyperspectral image. And then you see we map in the green and the red the two active pharmaceutical ingredients, which are smaller amounts. But just wanted to actually show you that a hyperspectral image contains spectral data in every pixel, and that data can be used to map the unique elements in the actual hyperspectral image. With that, we want to go back and show really quickly a food safety or food counterfeiting application. And in this application, we're going to look at skim milk and melamine. And in this first PowerPoint slide, you see this is a hyperspectral image of skim milk powder control that we know has no melamine in it. And you see this is an example spectrum from a pixel of the skim milk powder. In the next slide, we're looking at pure isolated melamine powder. And we see in red the spectral response of the melamine. And we can see that it has a very different spectrum in terms of the spectral response from the skim milk. And knowing that these two are different, we can then look at samples that contain small amounts of the melamine in the skim milk. And here we have a sample where we have a mixture of melamine and skim milk powder together. Now, to our eyes, there's no way we could ever see the difference between these two. That's the reason why melamine has been used as a counterfeit element in baby formula and other things. However, the spectrum of every pixel, remember, differentiates melamine from the skim milk powder. So if we look in this image here, in red, we're mapping all of the pixels where we have melamine based on its optical spectrum on a per pixel basis to demonstrate that 3,656 of the pixels in this image are mapping for the melamine. So even though our eyes can't see the melamine, the spectral analysis in the image can show it. And here we're just showing this, at a more magnified image, showing the melamine in red and the non-melamine skim milk areas here in white and black. So this is a very brief video just to show how hyperspectral microscopy can be utilized for drug characterization and for food characterization, looking for unsafe or counterfeit items in either of those two examples. If you would be interested in learning more, you can just send us a quick email at info@cytoviva.com, and we'd be pleased to talk about samples or different types of foods or drugs that you might be interested in learning about as it relates to this technology. Thank you.